Welcome back to this next video in the series of lectures on the cell cycle. In the last video, I've told you that in the G2 phase of the cell cycle, we are increasing the concentration of a specific cyclin known as the cyclin B. And the cyclin B was interacting to its specific CDK that was known as the CDK1. And the complex of the cyclin B and the CDK1, that was actually the MCDK. So during the G2 phase, the cyclin B and the CDK1, they were combining with each other, but the CDK1 was not getting activated because at the G2 phase, different kind of the proteins, they were interacting with the CDK1, actually making them inactive during the G2 phase of the cell cycle. One of the protein that was interacting with the CDK1 was the CDK activating kinase, or the CAC protein, and this kinase was phosphorylating the CDK1 at the position number 161, and this phosphorylation was actually stimulatory in nature. But at the same time, this CDK1 was phosphorylated by another enzyme, which was the V1, and this V1 was phosphorylating the 2 amino acid at the active site of the CDK1, that was threonine number 14 and tyrosine number 15. And the phosphorylation of these two sites, that were actually inhibitory in nature. So in the M phase of the cell cycle, what we were doing is that we were activating a phosphatase, and that was the CDC25. Initially, the CDC25 was in inactive form, and another kinase, the polo kinase, that was actually going for the phosphorylation of the CDC25, partially activating the CDC25. By that I mean that this CDC25 was going to remove the inhibitory phosphates from some of the MCDK, and some of the MCDK, they were actually getting activated. So once some of the MCDK, they were activated, they were going for the phosphorylation of the CDC25, and the phosphorylation of the CDC25 by the MCDK were fully activating them. Thereby they were activating all of the MCDK available in the cell. The MCDK at the same time was also phosphorylating the V1 protein, and this phosphorylation was inhibitory for the V1. So you are actually activating the MCDK at the M phase of the cell cycle. Now, once you have activated the MCDK, what this MCDK is going to do? This MCDK is doing a lot of functions at the start of the M phase, and one of the important function is the condensation of the chromosomes. Now, this chromosome term is actually used for the condensed form of the genetic material. So this would be like the condensation of the chromatin, or the condensation of the genetic material. The condensation of the chromosome is not an appropriate terminology. So at the start of the G1 phase, you are actually going for the condensation of the chromatin material. How the MCDK is going for the condensation of the genetic material then? This MCDK, being a kinase in nature, is going for the phosphorylation of a protein complex known as the condensin complex. And in eukaryotic cell, you are going to find two kinds of the condensin complex, and both of them, they are involved in the condensation of the genetic material. One complex is known as the condensin 1, and another complex is known as the condensin 2. And both of these complexes, they are having 5 subunits. Now, 2 of the subunits, the SMC2 and the SMC4, they are present in both of the complexes, the condensin 1 and the condensin 2. And if you remember, I've told you this SMC is actually the structural maintenance of chromosomes. So here you're talking about the structural maintenance of chromosome 2 and the structural maintenance of chromosome 4. You have already seen the SMC1 and the SMC3 proteins in the cohesion complex that was actually interacting with the sister chromatids, keeping them attached to each other. So this is one of the important function of the MCDK, that by phosphorylating the condensin complex, it is going for the condensation of the genetic material. Another important function of the MCDK is to break down the nuclear envelope, because if you are not going for the breakdown of the nuclear envelope, you will not be able to attach the microtubules to the sister chromatids. You cannot actually separate them into 2 daughter cells. So how the MCDK is going for the breakdown of the nuclear envelope. If you look at the nuclear envelope, the nuclear lamina is underlying shell of the polymerized lamin filaments. These polymerized lamin filaments is actually giving the nuclear envelope its structural rigidity. Now, if the lamin filaments, if this polymerized lamin filament is removed, you can actually disturb the rigidity of the nuclear envelope. You can actually break it down. This is what is happening in the M phase of the cell cycle, at the start of the M phase of the cell cycle. Now, the MCDK, that is going for the phosphorylation of the lamin proteins, and when these lamin proteins, they are phosphorylated, they are actually depolymerized. So once you have depolymerized a very important structure of the nuclear envelope, the nuclear envelope actually breaks down. That is going to be broken down into pieces. Another important thing about the nuclear envelope is that the MCDK, they are also going for the phosphorylation of the nucleoporine polypeptides. Now these are the protein structure that are present in the nuclear envelope, and they are actually responsible for allowing the things to move in and out of the nucleus. So the MCDK, when they phosphorylate this nucleoporine complex, these nucleoporine polypeptides are removed from the nuclear envelope. It is also going for the degradation of the nuclear envelope. Another important thing that is going on at the very start of the M phase, and that is carried out by the MCDK, is the assembly of the microtubules. If you have broken down your nuclear envelope, that was for a purpose, and the purpose was to attach the microtubules to the sister chromatids. Now these microtubules, if you remember, I've told you, is made up of two proteins, the alpha tubulin and the beta tubulin. And the alpha and the beta tubulin, they were making the heterodimers, and then these heterodimers were getting attached to each other to give you a long polymeric microtubule. Like if you can see over here, this is one heterodimer, this is another heterodimer, this is another heterodimer, and so on. They are attaching to each other to give this long polymeric structure to you. If you talk about the structure of this or this polymer of the microtubules, it actually have got two ends. Now this minus end, if you can see this minus end, that is actually the alpha tubulin end, and this particular end is attached to the gamma tubulin ring complex. And if you remember the machinery of the cell cycle, we have discussed that in detail. So if you have any problem, you can revisit the machinery video of the cell cycle. So this alpha end is the minus end of the microtubule, and this beta end, that is actually the plus end of the microtubule. Now we are not talking about the mathematical plus and minus. Here the plus and the minus means that at the plus end of the microtubule, they are elongating, and at the same end they are going for shrinkage. While the alpha tubulin end, which is the negative end, no elongation and no shrinkage is involved, and this alpha tubulin end is actually providing a stability to this long polymeric structure. Now how the microtubules, they are going to elongate and how they are going to shrink them. Let us discuss this in a little bit detail, how the elongation and the shrinkage of the microtubules take place. If you look at a single heterodimer, like you have got an alpha tubulin, you have got a beta tubulin. Now this beta tubulin, it have got the ability to bind to two molecules. It can bind to GDP, by that I mean the guanine nucleotide diphosphate, but it can also bind to the protein of GTP, which is the guanine nucleotide triphosphate. And when the beta tubulin binds to GDP, it have a different meaning, and when it binds to GTP, it have got a different meaning. Now, when the beta tubulin that is attached to the GDP, in this particular form, if you look at the binding angle of the alpha and the beta tubulin, that is 12 degrees. and when the attachment of the alpha and the beta tubulin is 12 degrees, it is a relatively unstable state of this heterodimer. And if the heterodimer is unstable, like if you are making this heterodimer unstable, that means you cannot go for the polymerization of the microtubule because the subunits, they are unstable. Now, if you want to polymerize this, the first thing you have to do is you have to stabilize these heterodimers, these building blocks of the microtubule, so that you can make a long polymer. In doing so, what you have to do, that you have to remove the GDP from the beta tubulin and you have to attach a GTP to the beta tubulin. Now, when the GTP that is attached to the beta tubulin, you can see that the attachment angle of the alpha and the beta tubulin, that is now 5 degrees, a very stronger kind of the interaction. And when the alpha and the beta tubulin, they bind strongly to each other, you get stronger subunits, and these stronger subunits can actually combine to each other to give you a long polymeric microtubule. Now, how the beta tubulin, how the GDP of the beta tubulin is replaced by a GTP. The replacement of the GDP of a beta tubulin by a GTP requires the action of another enzyme, which is known as the guanine nucleotide exchange factor. For short, we call it as the GEF. So what the GEF is doing, that it is going to remove the GDP from the beta tubulin and it will attach a GTP to the beta tubulin, and by doing so it is making these heterodimers stronger, and it is actually going for the elongation of the microtubules. Now, if you are interested in the shrinkage of the microtubule, what you have to do is that you have to remove the GTP from the beta tubulin and you have to attach a GDP to the beta tubulin. Now, that function is achieved by another enzyme which is known as the GTPase activating protein, or for short, you can call it as the GAP. So, if I summarize this, the GEF is going to replace the GDP with the GTP. The GAP protein is going to replace the GTP by the GDP. In both of these events, the attachment of the beta tubulin that is happening on the beta tubulin, the attachment of the GTP protein that is also happening, the GTP, not protein, this GTP molecule that is also happening on the beta tubulin end. Now, at the start of the M phase, we are interested in the elongation of the microtubule, because these elongated microtubules are going to attach to the sister chromatids, and when they are attached to the sister chromatids, they will be leading to the separation of the sister chromatid to the opposite poles. Now, what the MCDK is doing, that it is going for the activation of this set of proteins, and when they activate the guanine nucleotide exchange factor, they are going to remove the GDP from the beta tubulin, replace them with a GTP, thereby elongating the microtubules, and they will be able to attach to the sister chromatids. Another important function of the MCDK is that is going for the condensation of the gene transcription. We will discuss about this dynamic instability in a while, but let me tell you that how the MCDK is going for the cessation of all gene transcription. We have already discussed that the condensing proteins, they get activated, and the condensing is going to convert the chromatin into the condensed form of the chromosomes. Now, if you normally see that on the promoter region of a particular gene, if you are interested in the transcription of this particular gene, you are going to attach certain transcription factors to this promoter region, and when the transcription factors, they are attached to this promoter region, the RNA polymerase then attaches to the promoter and it leads to the transcription of the genes. Now, at the start of the M phase, as you have condensed the genetic material, that is going for the cessation of all gene transcription, because in the condensed form, the transcription factors are not able to bind to the promoter region of a particular gene. And if the transcription factor are not able to attach to the promoter region, the RNA polymerase will not be able to bind to this promoter. And if the RNA polymerase is not able to bind to this promoter, you cannot actually go for the transcription of the gene. So by this particular way, the MCDK is also going for the cessation of all gene transcription. Now, if you talk about the microtubules, they exist in a state which is known as the dynamic instability. Now, by dynamic instability, I mean that they coexist in the assembly and disassembly form. The dynamic instability refers to the coexistence of the assembly and disassembly at the ends of the microtubules. And as I've told you that if the beta tubulin is attached with the GDP, that is going for the disassembly, and if it is attached to the GTP, that is going for the assembly of the protein. If you talk about a single microtubule, what people usually think is that the microtubules, they are a single polymer of the alpha and the beta tubulin. But if you look at the tubular arrangement of a microtubule, they are made up of 13 protofilaments. Like this is one polymer of the alpha beta heterodimer. This one is one polymer. This is another polymer of the heterodimer, another one, and you have got 13 of them. So the 13 protofilament, they are actually arranged in a tubular form to give you a single microtubule. Now these protofilaments, they are attached by the heterodimers, they are attached to each other, but at the same time these protofilaments are also attached to each other like this, and they are also attached to each other. And in this particular form, they are actually giving you a very stronger kind of the structure, because they have to play a very important role of separating the sister chromatids equally between the two daughter cells. We'll continue this discussion in the next video.